Hi everyone! Welcome another year to the European Researchers' Night, a scientific outreach event that takes place every year all around Europe at the same time. The objective of this initiative is to explain to the society the research that it has been doing in the different centers in a way that everyone can understand the main idea of the topic. Some of the activities that you can join during the European Researchers' Night are talks, games, activities and shows. My name is Clua Villas and I will talk about therapeutic oligonucleotides. But first, let me introduce myself. I did my pharmacist bachelor in the University of Barcelona and, because of being a passionate of the laboratory and research, I decided to apply to do a PhD at the Institute for Advanced Chemistry of Catalonia in Barcelona, in the field of biotechnology and nanomedicine. This is my third year looking for different ways to use nucleic acids. Since now, I have been using them as a chemotherapeutic prodrug and as a nanostructure to carry drugs and targets to increase the internalization into specific body structures. Moreover, last summer term, I did an exchange in Munich at the LMU where I was introduced in the use of aminopeptide structures as a formulation to introduce a wide range of therapeutic nucleic acids. So let's go! Here you can see the topics that I'm going to explain today. First of all, what's a therapeutic oligonucleotide? It's a drug based on the nucleic acid chemistry. We use synthetic DNA or RNA letters to create a sequence. This sequence could be the drug itself or a system to deliver other drugs. Every letter has exclusive properties, so depending on the order that you put them, you can create unlimited therapeutic possibilities. The most important fact is that synthetic oligonucleotides can modulate processes in your body that are not possible to reach with the chemical conventional drugs. The main problem is how to introduce it inside the cell. And what happens inside the cell? This is a schema of a cell. The nucleus is the place of the cell that contains all the information necessary. This information is saved in the DNA. The humans have around 3 billion DNA bases. When we need something, the part of the DNA that contains this information is transcribed to RNA. The RNA molecule is huge and has parts that modulate how to read the containing information. So, once the cell knows what it needs to do, throw away these instructions and create the messenger RNA. The mRNA travels from the inside of the nucleus to the cytosol. Here it finds a lot of different ribosomal forms that translate the information of 3 letters to an amino acid. This amino acid joins the previous one, creating a risband. The ribosome stops adding new amino acids when it finds 3 letters that are translated to a stop signal. It's named peptide if it's small and protein if it's large. All the amino acids in the sequence interact each other, forming a 3D structure, which is the one that develops the correct function in the body. Types of therapeutic oligonucleotides. Here we have an overview of where the different types of existing therapeutic oligonucleotides develop their effect. Inside the nucleus of the cell, it takes place the action of Criscus, known as the gene editor, and the main action is change or delete a specific site of the DNA. The splicing switching oligonucleotides have the main function of modulating the information that will be present in the mRNA. In the cytosol takes place the action of the other therapies. The antisense oligonucleotide and the short interference RNA, the objective is the degradation of the mRNA through different pathways. And in the mRNA vaccine, best known as the COVID vaccine, they use it to synthesize new peptides that will activate the immune system versus future dangerous infections, or to supply a deficiency. antisense oligonucleotides, or ASO, is a single-strand sequence of 8 to 50 letters that is bound to the mRNA by complementary base pairing. This DNA-RNA complex activates the RNase H that degrades the mRNA. Another mechanism of action is by steric block. The double strand can't go inside the ribosome, so there is no translation. In both cases, the protein formation is sabotage. The splicing-switching oligonucleotides, or SSO, is a single-stranded oligonucleotide, but the structure has no negative charge. The main use of them is when a mutation is located in the RNA that produces an erratic protein that derives in a disease. So, the SSO is linked to the target sequence that we want to omit. This produces a changing on the reading schema, obtaining an mRNA without part of the information, but we will have a functional or half-functional protein reverting the disease. Short interference rRNA, or siRNA, mechanism is more complex than the before mentioned. In this case, we start with a long double-strand RNA with or without a sharp at the end. This is detected by the dicer protein that cut it in small pieces, obtaining the siRNA. When the siRNA is attached to the risk protein complex, one of the strands is removed and now the complex is activated. This association starts looking for complementary mRNA, and once it's detected, starts the enzymatic degradation of the mRNA. The interesting point is the large turnover of the activated risk complex, which is continuously looking for other complementary sequences, prolonging the degradative effect. The Criscus Therapy is a system composed by the guide RNA, around 20 nucleotides, and the Cas9 protein that is a Jan scissor. So, when the gRNA is bound by complementary base pairing to the target DNA region, the Cas9 protein cuts the DNA, deleting the erratic part. Once this is done, the natural mechanism prepares the DNA linking both sides. Otherwise, there is the possibility to introduce a correct DNA piece to replace the ones that was deleted before. Last but not least, the Mrna vaccine. In this case, the full mRNA containing the information about the protein of interest is kept inside a lipid nanoparticle, like an oil drop. This nanoparticle is introduced inside the cell and the mRNA is read by the ribosome and the protein is formed and delivered outside the cell to the full body. But the point is that this protein has a tag that stimulates the immune system and the body produces antibodies against this protein. So, if something with the same proteins get inside the body, the immune system will detect it and destroy it with the antibodies formed beforehand. Another possibility is to use the same system without the alarming tag to produce the protein to compensate a deficiency. Therapeutic oligonucleotides on market. Here you can find a table with 14 oligonucleotide-based therapies available on market. The first one, Vitraven, was accepted in 1999, and the last one, Inclisiran, was accepted at the end of 2020. That's all. Let's end this talk with a few concepts to keep in mind. Oligonucleotide therapy can change your whole DNA information, and it's height specific. It uses the natural pathways in the cell to produce the effect. It can eliminate a misspelling sequence or stimulate the immune system. With the oligonucleotide therapy, we are able to do a specific drug for a single patient. Thanks everyone for watching this video. I wish you have enjoyed it and has learned something new about these novel therapeutics. Before closing the browser, have a look to other short talks in the YouTube channel of la Nit de la Recerca and the schedule at the website lanitdelarecerca.cat to be aware of all the activities. Stay safe